This is where we're going to move from a graded to an action potential at this point. So your action potentials, you'll see these referred to as an all or none phenomenon. They're either going to occur as a maximal depolarization. If you reach that trigger zone and it fires, it goes, right? Or if you don't reach that trigger zone, it doesn't go, right? All or none. It either goes or doesn't go. If you get above, say, -50, then you're going to go. If you don't, nothing. Not a graded one, not a big one and a small one, nothing. Right? And if it's a really big graded stimuli, it's going to cause this action potential. If it's a much smaller stimuli, but enough to do the trigger zone, it's going to cause the exact same action potential. All right, so the strength of the graded potential doesn't have any amplitude. It's always going to be the same dynamics, membrane dynamics of this action potential. And what we'll see is this wave, this just like we saw with the muscle cell, right? This wave of excitation going down here, right? And instead of diminishing, right? If this was a graded potential, right? You'd have this, and this, and then this, and then this, and then this, right? But this is an action potential. And each one of these ones, little local depolarizations, this flip-flop of voltage, membrane voltage, right, is going to be the same here as it is over here. That's your action potential. And this is all going to be due to voltage gated channels, right? As we will see. And so over here, you just had that current flowing, the sodium ions or whatever flowing inside the cytoplasm and outside here, right, based from that initial point of stimuli. At some point over here, that current flow is going to create an opening of these voltage gated channels. It's going to go down the length of the axon. So just to kind of this again, your book doesn't really do this too well, but I want to mention that threshold stimulus at the axon hillock around there is basically the equivalent of what your sodium channel needs to open when it gets a stimuli, right? So at -70 over here, your voltage gated sodium channels are closed. Sodium does not get in the cell, right? Just like, we want to keep sodium out. At a certain depolarizing stimulus, if it's enough, which is about 55, again, your book doesn't specify these, but sometimes it helps to kind of know the numbers, right? -70, and all of a sudden you've got to 55. At that voltage, this opens up this little channel over here called the activation gate. Right, it opens up, which allows sodium to go inside the cell right here. So it's voltage gated. When it senses that it's depolarized to -55 millivolts, it opens up. So you're already depolarized, and now all of a sudden that depolarization is going to cause this further influx of positively charged ions. So what's that going to do? Right? It's going to vastly, massively increase that depolarization. So you're going from -55 in this one picture to about 0. Right, so sodium is flooding in, and the cell is getting more and more positive. At some point, you had this activation gate open due to voltage. At some point, you have this second gate down at the bottom right here, which is going to close. And this is going to close, and sodium is going to be blocked again, even if this activation gate is still open right here. Sodium was flowing in, and at some point during this, based on either time or voltage, right, that inactivation gate closes, and all of a sudden sodium influx stops somewhere around +30, right? Now your cell's gone, this area has gone from being -70 to a +30 on the outside right here. So you have this positive charge on the outside. I mean, you had a negative charge on the outside relative to the inside, positive on the inside, and now sodium is blocked. And then we start repolarizing at this point. So this repolarization, as we'll see, is going to be caused by potassium leaving it, right? But at this point, now the sodium channel is back. Once it's repolarized, got back to -70, everything is ready for action again. So that's what's happening with this sodium gate. So all these voltage gated sodium channels all along your axon right here, right? Starting at the trigger zone right here. You had a graded potential. It reaches this area, depolarization event. Apparently, it was above that voltage that opens up that activation gate of the sodium channels, and it lets all the sodium in, and this yellow region over here is indicative of all the sodium flowing in, become a positive to charge, whereas the blue part is still negative. All right, so voltage gated channels are opened up, sodium enters, right? And it's going to do that kind of depolarization thing, just like your regular graded one, except that, you know, this local current flow, this depolarization is going to open up more sodium voltage gated channels that are over here. All right. So that is, you know, these voltage gated channels are going to move in this direction right here. You have voltage gated channels everywhere, but we'll see the dynamics of both the opening and closing of the sodium, as well as the opening and closing of the potassium channels, are going to cause this area back here, right? This loss of potassium. What happens when you lose potassium? You become more negative, right? So following this wave of depolarization, you're going to repolarize with this potassium outflux, and that's going to prevent it from going backwards. That's why it's going to move this way. This is going to be called the refractory region over here. And this is the inactive region where active region is moving toward, but following behind it, you're going to have this outflux of potassium, bringing the cell back toward resting potential. Now we're going to look at this in a couple different graphs, so you get the idea, but that's what's happening down here. Here's that kind of graph again, right? We're following the voltage change as a matter of time, right? This is time as they're recording this, right? So here's the stimuli sometime over here, right? The resting membrane potential, again, -70. There's a depolarizing stimulus. It's around -55, which opens up those sodium channels. Influx, right? Rapid sodium entry depolarizes that all those positive charges come in here, raises it until it reaches about +30. And then those sodium channels close, right? So it doesn't go up higher. But there's something else going on, not shown right here. The potassium channels have opened, right? But they didn't open at the same time. Here, they opened up a little bit later. So there's this sequence of events here, but I want to show you what's going on with these, because there's two things that are contributing to this curve right here. And they have to do with the opening and closing of the potassium and sodium channels and the differences in how fast they do it. So again, back to my coronavirus, looking at daily deaths, right, over the course of last year. As a whole, if you look at the United States as a whole, you see this big spike, right, in deaths. And then it went down as summer approached. And then you saw another spike in deaths as the US, right? This is one big curve that accounts for the entire country. But if you looked at any one particular region of the country, right, you'd see how this graph is made, right? So over here, this chart is something like Maryland, somewhere on the East Coast. During April, you saw this huge spike on the East Coast, right over here, of deaths right here, which went down by summertime, right? At that same time, there was really nothing going on in the Sun Belt. Florida, Texas, Arizona didn't have that many relatively deaths right here. But as summer approached and these guys went down over here, you saw this big spike over here as the virus kind of spread through those areas over here, right? So some people call this the second wave. It was really the first wave going through this population. And then for the second one, it was kind of the whole United States was getting this winter wave over here of deaths right here. But you can see these are very different. If we look at certain areas, and you could look at any one particular, New York, all that, Massachusetts, the really East Coast cities were a little bit further, they were a little bit bigger, right? But they were all unique. When you put them all together, right, over here, this is the United States, the darker curve, but it's a conglomerate of your East Coast ones, which are really high, your Sunbelt ones where not much was going on, right? So it's kind of a combination of these two. And then the second spike is due to, and almost entirely due to Florida and Texas and stuff, their spike, whereas the East Coast had already gone down. So it's somewhere in between there, right? So this individual spike is made up of a whole bunch of different individual things going on. This is an aggregate. And it's going to be the same for our spike, right? When we look at that action potential, there's different channels contributing differently. So here's the difference. Here's the voltage one that we're looking at, right? There's a big rise in that depolarization going from -70 to +30 over here, and then drops back down. The contribution of the sodium channel, right, is pretty much linear. It's pretty much all due to sodium up to this point. What's happening over here is that this is an outflux, right? So potassium is leaving the cell, and that's what's eventually bringing this voltage down over here, right? Bringing this down over here. So these are opening, sodium is flowing in, causing this depolarization. Because potassium is flowing out, that's bringing the cell back, right? As the sodium ions were coming in, it's already starting to kind of go down a little bit, right? And the dynamics of this, the fact that it's opening up slower and it's staying open longer, right, is also going to cause this so-called after hypopolarization effect. Right, so sodium's coming in fast, causing this big spike in voltage raise. But at the same time, the reason it comes down so quick is at the same time, the potassium channels opened up due to that same voltage change, but they have the opposite effect, right? And kind of bringing this voltage back down. And they're going to stay open such that the cell's actually going to go beyond normal resting membrane potential to -90. Right, so that's what's happening with these two channels right here. Sodium's flowing in, creating positive charge inside the cell. Potassium's flowing out and kind of dampening that positive charge and creating a negative charge. So there's the dynamics here, plus the direction of flow, right? Sodium flowing in, potassium flowing out. So here's another, so here's kind of a similar graph right here, right? You have the -70, both channels, sodium, potassium are closed. At the depolarization event, right? That graded one, it reaches its threshold, sodium channels open, causing this big spike over here for sodium. Right? During that period, potassium channels open, right? And then what do you have? Right at this point, sodium channels close, sorry, and potassium ions start bringing it back down over here, right? Because the potassium's flowing outward. After that period, right, it repolarizes, right? The whole cell membrane repolarizes. And actually, because this remains open, it dips below, right, your normal membrane potential over here. So that is what's going on with those channels when they are moving through here. Again, this is a different way of looking at it. And this whole feedback cycle, right? Depolarization, looking at the sodium part of it, is going to trigger this sodium channel opens very rapidly. Sodium enters the cell. There's more polarization, more sodium channels open up. They keep on open up. And as this is happening, though, at any one given region, the sodium channels inactivation gates are closing, causing the sodium to stop. But at the same time this is happening, these slow potassium channels are opening, potassium leaving the cell, and that's repolarizing it, right? Bringing it back toward -70. All right, so we'll break these down into individual steps right here as we go at any one particular area and looking at the chart right here, right? I'll close up. This is in your book. right over here at rest, resting state. Again, activation gate of the sodium channel is closed. Inactivation gate really doesn't matter at this point. We could ignore that. Your sodium channels, your voltage gated sodium channel gate is closed. Channel is closed. Your resting membrane potential, there is a buildup of negative charges, right, inside and equal on the outside roughly, right? Or it's going to be minus 70 overall, but there's that charge right there. All right, so this is a resting membrane. We have this depolarization phase. Membrane potential reaches threshold based on that graded one. Right, at that point, your sodium channel activation gates over here open, and sodiums move in. Sodium ions move in, and this is going to cause that big increase, this big spike in positive charge. So depolarize, right? Going up toward 30. The sodium inactivation gate around when it reaches about plus 30 or so, they're going to plug that up, stop the influx of sodium channel, right? But to really get it back down, you also open up your potassium channels have opened up, right? And what's that doing? There's positive charges leading, making this more negative. So these are becoming repolarized as potassium ions leave right there. And they start to build up back down, right, as we continue down. Potassium outflow continues, right, and a negative charge starts to build up. Right? And then once it's at a certain point, minus 70 or so or lower, then those potassium gates are going to close. They don't quite close at membrane potential though. This outflow of potassium, voltage gated channels, potassium channels remain open until they become more negative, right? So many have flowed out, right? This is what your so-called potassium equilibrium is, right? They keep on leaving the cell because it's because of a concentration gradient down here, right? And they flow out, right, until the cell becomes more negative, right? And as those close, then all your normal leak channels and ATP ACE pumps kind of bring it back to normal, right? So this is just another chart of a similar thing. You want to look at what is happening at each one of these phases, right? What's happening with the voltage gated things. I'm not actually going to ask you about absolute or relative refractory period when the nerve, you can either not fire or you need a larger stimuli and everything. Don't worry about that, but just look at what the channels are doing at any one point during each phase of this. All right, where's the ions going? Which direction are the ions going? And then what's that causing relative to your resting membrane potential, right? So moving away from it, going back toward it. Let's kind of get that down. Another clinical note here. There are certain things that block these voltage gated sodium channels, right? This thing's called tetrodotoxin. It's a big thing for neuroscience. They want to block a sodium channel from the pufferfish, right? They put them in there and it blocks that and you stop action potentials. So in that case, if that was in your, muscle cells or any motor neurons or something, you'd block all your thing and you'd be paralyzed, right? So that would be bad. And for sensory neurons, it would also block transmission of sensory stuff. So that's a neurotoxin because blocking those channels' ability to generate an action potential. The other one is, similarly, you have these local anesthetics, right? Drugs, they actually block it. You put it on there and they block those sensory neuron axons so that even if they're being stimulated, which would normally cause you pain, right, you basically block that stimulation over there with these local anesthetics, Novocaine, lidocaine, stuff like that. Right. And then the other thing is cooling also, because as we'll see, action potentials, they are slower when they are cool. So you can actually slow down the transmission. That's why putting ice on a wound or something helps relieve the pain. So in both cases, all those long distance nerve impulses are lost because there's no more action potentials. because the voltage gated sodium channels are now blocked. All right, that was the all or none, lose no strength signal as it travels down the axon, known as the action potential. And next time we're going to make that action potential faster by adding a myelin sheath. All right, we'll see you next time.